Hi everyone, in this video we're going to be covering the NetSolP version 1 model. So this is a model that can be used for predicting the solubility as well as expression levels of a protein. In this tutorial we're going to be covering how to use it on the Neurosnap platform. If you don't already know, Neurosnap is basically just a service where you can access all these different tools and models very easily as an academic or an industry individual. This model is very straightforward to use. It only takes one input, and it's the proteins that you want to predict the solubility and expression levels of in a FASTA format. So we're just going to paste them over here and hit run job. And this is going to take a little bit of time to run, but after it gets queued up and runs, we'll be able to see the results. Alright, so we are back with the results. As we can see, this hardly took any time at all with a runtime of about 1 minute. And since we only submitted 2 proteins, we have both of them over here in this table. So the two most important columns are going to be mean solubility and mean usability. So solubility is essentially a value ranging between 0 and 1 that's predicted by the model. It's a normalized value, so it doesn't actually correspond to any physical things. But generally speaking, values closer to 0 tend to mean like very insoluble, at least in cell types like E. coli, whereas values closer to 1 tend to mean like highly soluble. So anything above 0.75 can be treated as fairly soluble, at least in cell types like E. coli, according to this model. Usability, on the other hand, is a little bit more complicated and somewhat subjective. It's more of a combination between solubility and the expression of that protein within E. coli. But then again, expression is, can depend on a lot of different things like codon optimization, all sorts of stuff that is not taken into account by this model. So generally, this is not as important of a metric. As for these other columns, the reason why it says, solubility model 1, solubility model 2, 3, and so on, is because this service actually uses an ensemble of different models that all predict solubility and usability. Ensembles, or you can think of an ensemble as like having 5 different models, and each one gives its own prediction. And then once every single model has done its prediction, those results are aggregated and the average is presented over here. So these two columns are by far the most important, but in case you want to see what the other models think as well, we have the metrics available over here. So I hope this tutorial has been helpful. If you have any questions, leave them in the comments and we'll try to get back to you. And yeah, we hope to see you in the next video.